Welcome to another discussion. This is an important topic in the genetics, that is the crossing over to produce the genetic recombinations in the next generation. This is happening during the meiotic division, the reductional division. This crossing over or the production of genetic recombinations is the main way of evolution of organisms. We can say that number of flora and faunal biodiversity is living on this earth because of this crossing over or genetic recombinations during the meiotic division. That is actually happening during the sexual reproduction. This crossing over or genetic recombinations are produced in the reductional division in between the homologous pair of chromosomes. They can produce the different variations in the next generation. In this class, I am going to discuss about these. These are the questions I am going to. These are the questions I am going to solve. Those questions are: what chromosomes are actually involving in the crossing over? If the chromosomes are participated in the crossing over, what will be the product? Means, how do they produce the genetic recombinations? Some chromosomes are involving in the crossing over, some chromosomes are not involving in the crossing over. Then, what is synaptonemal complex? What is its role? So, what is DNA doubling and what is chromosomal doubling? So, what is chiasma frequency or what is percentage of crossing over? What are bivalents? How two tetrads are formed? What are endonucleases and ligases? What are the roles of these enzymes in this crossing over? Then what are the consequences of crossing over? Means how do these crossing over is useful for the organisms? These are the questions I am going to solve. At the beginning, I am going to discuss about the overview of the meiotic division or meiosis or reductional division. Meiosis division is containing meiosis 1 and meiosis 2. This meiosis 1 is important. This meiosis 2 is equal to the mitotic division. So during the meiotic division, the chromosome number will become half or reduced to half. Then meiosis division is just like the mitotic division without entering into the DNA replication. The cell which are formed in the meiosis 1 enters into the meiosis 2, but the main changes are taking place during the meiosis 1. That is, chromosome number is reduced to half, and means the cells participating in the meiotic division are containing 2n number of diploid chromosomes at the beginning. But after completing this meiosis 1, the cells form only one set of chromosomes. Means the chromosome number is reduced to half. Because they are going to form the gametes, the gamete should contain only one set of chromosome. The father can give one set and the mother can give one set. During the fertilization, they can form the diploid organism in the next generation. So, what is main aim of this meiosis 1 in forming the diploid state to haploid is maintaining the correct number of chromosomes in every generation. Another important thing during this meiosis 1 is crossing over, exchange of genetic material in between the homologous pair of chromosomes. If the crossing over take place, the genetic recombinations are produced. With those genetic recombinations, new variations are produced in the next generation. Those variations are raw material for the evolution of organisms. So, in this class, we are going to discuss about how do the crossing over is taking place during this meiotic division. So, here the meiosis 1 is containing the prophase, metaphase, anaphase, telophase and cytokinesis. The meiosis 2 is also containing the same steps. But in the meiosis 1, during the prophase 1, there are 5 important substages are there: leptotene, zygote, pachytene, diplotin and diakinesis. I am going to discuss about these 5 stages now, because the crossing over mechanism is tied up with 5 stages only. The remaining stages are discussed in meiotic division. Another video is there. The link is pasted in the description box. If you want to watch it, you can go and watch it. Now we have to stick to the crossing over. The father and the mother. Father is containing homologous pair of chromosomes, mother is containing the homologous pair of chromosomes. Actually, these homologous chromosomes present in the father are his parents. From his mother, he got one chromosome. From his father, he got another chromosome. She is also one from father, another from mother. These are the homologous pair of chromosomes. Those are 22 pairs autosomes. In each pair, one is from father, another one is from the mother. In the 23rd pair, that is XX or XY, mother and father. Okay. These are the homologous pair of chromosomes. The cell enters into the replication phase during the cell cycle. During the replication phase, that is called the S phase or synthesis phase, the DNA is doubling. So the DNA is doubling by the replication of DNA during the cell cycle at the S phase. So what is happening? This is already existing DNA and the new DNA is synthesizing and they are holding with each other. This is old DNA, this is new DNA, for example. So the DNA doubling means the DNA synthesis is taking place before starting the meiotic division. Then the cell now enters into the prophase stage. In the prophase stage, the first one is the leptotin stage. During this leptotin stage, the synthesized chromosomes which are containing the double DNA with their telomere region, they are attached to the laminal proteins. These are the laminal proteins. This rose color is showing the laminar proteins. Those are located on the inner surface of the nuclear membrane. That is a proteinaceous structure. This proteinaceous structure, laminal proteins, are used to hold these chromosomes to it. At the telomere region, these chromosomes are attaching to these laminal proteins. So why they are attaching to the laminal proteins? What is the? Because the homologous pair of chromosomes, so while they are coming with each other, to avoid the unnecessary movements, these are attached to the laminal proteins. So these are attaching at the laminal proteins at the telomere region. Here you can see this is the inner membrane that is containing the laminal proteins. These are the chromosomes attached to the laminal proteins to avoid the unnecessary movement. So now actually each chromosome is containing two chromatids, but they are not clear because they are highly coiled structures. Now the second stage is the zygote stage. Homologous chromosomes are pairing, that is called the synapsis. The important step is the synapsis taking place during this stage. These two are the homologous pair of chromosomes. In this each homologous chromosome, there are two strands which could be called as the chromatids next. So these are the two homologous chromosomes are coming close to each other, moving close to each other. These are now calling as a bivalents because they are containing two. These are called bivalents. This phenomenon, what is that? Coming of homologous chromosomes to the close is called the synapsis. That is happening during the zygote stage. But how do the chromosomes are able to come close for pairing? How they are coming close to each other? This is an important thing to be discussed. Here is one important structure is forming in between these bivalents, that is the synaptonemal complex. This is the synaptonemal complex which is made up of some structural proteins and enzymatic proteins, DNA and RNA molecules. The combination of these four is made the synaptonemal complex. This synaptonemal complex is containing one lateral line. This is also another lateral line which is showing in big shape. But in between these two lines, there is a midline is there. In the midline, there is one structure is there. This is the recombinant nodule which is containing enzymes which could be used for the crossing over. That is discussed next. This is the synaptonemal complex. This synaptonemal complex is mainly used for this purpose, segregating chromosomes from the other chromosomes. In the nucleus, many chromosomes are there. There are many chromosomes are present in the nucleus. Each homologous pair of chromosomes containing their own synaptonemal complex. the synaptonemal complex of this homologous chromosome, these two homologous chromosomes are containing another synaptonemal complex. So every homologous pair of chromosomes are containing the synaptonemal complex. If the synaptonemal complex is present in between the homologous chromosomes or bivalents, they can hold to avoid the unnecessary movements. Means other chromosomes cannot mingle these chromosomes. To avoid this contamination, this synaptonemal complex is used. And also this synaptonemal complex also pulling the chromosomes to close for synapsis. Then continue the pairing till completion of crossing over. During the zygote stage, actually this synaptonemal complex is formed. This synaptonemal complex is maintaining the integrity and originality and structure of these two bivalent chromosomes. In Pachytene stage also, till the completion of crossing over, this is maintaining the synaptonemal complex is maintaining the structure of these two bivalents. We will see. Next stage is the Pachytene stage. The important stage this is, because main two important things are happening at this time. One is the duplication of chromosomes and the other one is the crossing over. The duplication of chromosomes, remember this point at the beginning. Yes, this is duplication of DNA, means DNA is doubling. During the cell cycle, at the time of S phase, the DNA is replicating and the DNA is doubling. That is called the doubling of DNA or duplicating of DNA. Now this stage, during the Pachytene stage, this chromosome is containing two arms or two strands. These are called as the chromatids. These are now separating, just separating, only physical change. These are separating, that's why it is called as the duplication of chromosomes. While they are attaching with each other, that is totally called as one chromosome, but now if they are separated, those should be called as the chromatids. Next they will be called as the chromosomes. So at this time, these are called the duplication of chromosomes. The duplication is started at the end telomere region and extended towards the centromere region, like this. At the telomere region, that is started, that is completed at the centromere region. In this direction also, like this. Zipper-like manner, opening of zip. So these are the unsplit chromosomes. After splitting the chromosomes, these are showing like this. This is the centromere region. These two arms or two strands are called as the chromatids now. But later they will be the separate chromosomes. This separation of chromosomes is a zipper-like manner. This separation is like this. In mammals, the separation is started at the end towards the centromere region, like this. But in some other organisms, this is observed that the separation, the zip opening is started at the middle region, that is the centromere region, and extended towards the telomere region and ends of the chromosomes. As a result of this separation or doubling of chromosomes, two sister chromatids are formed. These two are the sister chromatids. These two are the sister chromatids, but these two are called the non-sister chromatids. These two are called the non-sister chromatids. The synaptonemal complex is present in between these chromosomes also. Now, this bivalent formed two chromatids. This bivalent is also formed two chromatids. Now, this is containing the four arms or four strands chromatids. This is called as a tetrad. At this stage, another important phenomenon is the chiasma formation. Chiasma means attaching the non-sister chromatids at a particular region. That is the chiasma formation. This chiasma is formed at the recombinant nodule. Synaptonemal, this is the synaptonemal complex. At the middle line, the recombinant nodule is present, that is containing different enzymes. At this region, actually, the chiasma is forming. Means at this region, the crossing over is going to take place. the crossing over between non-sister chromatids. These two are the non-sister chromatids. In between the non-sister chromatids, the chiasma is forming. The crossing over is going to take place. So during this stage, after the chiasma formation, these chromosome segments are cut and exchanged. That is called crossing over. During the Pachytene stage, the crossing over is also completed. But there is somewhat big mechanism is there during this crossing over is synaptonemal complex first pairing the chromosomes are synapsis. Then the recombinant nodule is formed, the chiasma holding the chromatids. The recombinant nodule, this is the recombinant nodule. At this stage, the two sister chromatids are coiling with each other. That is called the chiasma formation. Then the chiasma, after coiling, this chiasma can hold these chromosomes. This chiasma is maintaining that coiling. So next, the recombinant nodule is containing this specialized enzyme called the endonuclease enzyme. This endonuclease enzyme can cut the DNA at specific points. Then recombinase enzyme present in this recombinant nodule can facilitate the exchange of cut segments, exchanging. Then after the exchange, the ligase enzyme produced from this nodule is annealing or pasting these exchanged segments. Then the crossing over is completed. Means synaptonemal complex proteins are pairing the homologous chromosomes. Then after doubling the chromosomes, the recombinant nodule can produce enzymes. Endonuclease is cutting, recombinase enzyme is exchanging the segment, ligase enzyme is re-annealing or pasting the chromosomes. Then the crossing over is completed. Now the recombinant chromosomes are produced. Means after completing the crossing over, recombinants are produced. Then some of the questions are also remaining there. Chiasma frequency. What is chiasma frequency? What is the percentage of crossing over? Means showing that at two regions the chiasma are forming or crossing over is taking place. Does all the chromosomes are paired at two regions or more regions or one region? Means how many chiasma are forming, how many places the crossing over is taking place. That is the question of chiasma frequency and percentage of crossing over. How many chiasma are formed in a pair of homologous chromosomes? How many crossing overs are taking place in it? That is the main question here to be discussed. This is single chiasma formation or single crossing over. This is double crossing over. In this double crossing over, these two non-sister chromatids are crossing here. Those same non-sister chromatids are crossing here. This is double crossing over. But other type of double crossing over is this. These are 4 chromatids. In these 4 chromatids, this first chromatid, which is showing red color, is not involving in the crossing over, but remaining this one, this one, this one. These 3 are involving in the crossing over. They are produced recombinants. This is 3-strand double crossing over. But whereas in this case, all the strands are involved. All the 4 strands are involved in the crossing over. They formed 4 recombinants. This is 4-strand double crossing over. So the crossing over may be single crossing over or double crossing over. In case of double crossing over, 2 chromatids may be participated, or 3 chromatids may be participated, or all the 4 chromatids may be participated. So what is determining actually this? This is the question. Means the question is how many chiasma are forming or percentage of crossing over. Means we have discussed kinds of crossing over. Single crossing over or double crossing over. In the double crossing over, different types. What are the causes for these different types of crossing over or chiasma formation? Finally, they concluded that for the reason for the percentage of crossing over or chiasma formation, frequency of chiasma formation is the length of the chromosome. the length of the chromosome and distance in between the genes. This is already told by T.H. Morgan in his chromosomal theory of linkage. So the length of the chromosome and the distance in between the genes located on one chromosome are determining the number of chiasma formation and the crossing over. How this is means, if the chromosome is long, there is a more chance for many crossing over. So if the length of the chromosome is long, many chiasma are formed. If the chromosome is short, very less number of chiasma are formed. Means if the length of the chromosome is high, the crossing over percentage is also increased. If the length of the chromosome is decreased, the crossing over percentage is also decreased. Means the length is directly proportional to the chiasma formation or crossing over. This is directly proportional. Next one is the distance. The chromosome containing the genes, the distance between these three genes is less, but the distance between these three genes and the fourth gene is very wide. So here is a chance for crossing over. Means if the distance between the genes is wide, there is a chance for the formation of chiasma or crossing over. If the distance is very less, those genes are linked with each other, there is a less choice for the crossing over or chiasma formation. So means the distance is directly proportional to the chiasma formation and crossing over also. If the distance in between the genes is increased, more chiasma are formed, more crossing over is taking place. If the distance is decreased, if the distance is very less, that means what? The genes are linked with each other, hold with each other, are coupled with each other. They do not want to separate it. Then the chiasma formation and crossing over percentage is decreased. So this length of the chromosome and the distance in between the genes are determining the percentage of crossing over. And what are the consequences of the crossing over? Then see, at the beginning I was discussing about the biodiversity of this earth. Rich biodiversity, fauna and floral diversity is living on this earth. This much of floral, faunal and bacterial diversity is the product of crossing over. If there is no crossing over, no variations are produced in the next generation, no new species are produced in this nature. That is we will discuss in the evolution. Okay, then we will see what are the consequences of crossing over. Is crossing over take place in between the chromosomes, then genetic recombinations are produced. With these genetic recombinations, next generation are containing some variations. Those variations are good variations are selected by the natural selection. Those characters are given to the next generation. Those are the adaptations take place in the next generations. Those adaptations can be from the new species after many generations on this planet. That is the beauty of crossing over. If two crossovers occurs between two strands, this will result in this chromosome and these two chromosomes. If two crossovers occurs between same two strands, these are the strand, one crossing over here, in the same strands another crossing over is taking place here. As a result of this, what type of segments are separated? That is the question here. In this case, the middle segments are separated. So exchange of middle portion in between these. That's why we have discussed about the single crossing over, double crossing over. In the double crossing over, 3-strand crossing over and 4-strand crossing over. This is also discussed in detail in another video, that is the kinds of crossing over. Double crossover involving dash strands result in 100%. Means if the 100% crossing over should take place in this, what chromatids have to be participate in the crossing over? all the chromatids have to be participate to get the 100% recombinations. So that is called as the 4 strands have to be participated in the crossing over to produce the 100% recombinations. That is 4 strand double crossover. That is this one. 4 strands are involving, 4 genetic recombinations are producing. This is 4 strand double crossing over. The another question is, alleles of different genes that are on the same chromosome may occasionally be separated by a phenomenon is called. The genes located on one chromosome called the linkage group. Those because those genes are linked genes, but sometimes they are separated, involved in the crossing over and form the new genetic recombinations. That is the answer. Genetic recombination occur through mitotic fertilization? No. Mitosis and meiosis? Both cannot. Meiosis and fertilization? Yes. This is answer. Crossing over occurs in the, at what substage of the prophase the crossing over is actually taking place? Direct answer is the Pachytene stage, leptotin stage for the structural arrangement, and zygotine for the synapsis, Pachytene for the duplication of chromosomes and the crossing over. Then another question is, what is the unit of crossing over? Actually, where does the crossing over is taking place? Actually, a gene contains a cistron, muton and a recon. Different places are present in it. At this place, at the recon place, the recombination is taking place. Means at this place the crossing over is taking place. Linked genes are separated by, okay, if the genes are linked, those should be separated by crossing over. That is the direct answer. This video is prepared for those who are preparing for the higher level examinations, junior lecturer, degree lecturer or some other competitive examinations. So the important points are discussed. You can practice more questions, then you can get the more perfection. Then you can write your doubts in the comment section. Then I can answer those all questions. Thank you.